Yeah, fantastic. So I don't know where I mean I don't know if Parham has a an agenda or where you want to start. And maybe we can work from, just the way we have in this preamble, from the sort of, molecular redundancy to the bigger picture, and maybe end up with the information story.
I'll give you some history. And that is that I was a graduate student at Harvard back in the late 70s, early 80s. This was a time when there was huge debates going on about evolutionary theory, mostly about evolutionary theory for cooperation, for altruistic behaviors, and so on. This is when the social instincts stuff happened. In fact, I was taking classes and having conversations with many of the major players. Ed Wilson, Stephen Jay Gould, Dick Lewontin, Irvin DeVore. Then, interestingly enough, for 1 year, Bill Hamilton came to give a series of lectures. Bob
Mhm. Yeah.
Trivers and Bill
Mhm.
Hamilton gave a class together, which, a whole bunch of us just sat in on it. These guys personally are like oil and water. They're totally different personalities. They go at their theories totally differently. So Hamilton would start talking to us and turn to the blackboard and just be writing and talking at the same time. We never saw his face again. And the board was filled with equations, of course. Trivers, this was a kind of a theater-like structure, which they had a blackboard and a stage. Trivers would sit on the front of the stage, kick off his sandals and free associate. And so we alternated between these two. It was just a great conversation that happened. And then, of course, all of us in the audience were out, we're trying to put it together.
Mhm.
But the problem that was trying to be answered then had to do with synergistic relationships and how they develop. This was, of course, a decade, a little more than a decade after Ono's theory, and nobody was paying attention to it at all. And in part because there was also a lot of fear about the Kimura story of gene
Sure.
neutrality, that maybe we shouldn't have a theory of neutrality. That can't, so there's big arguments about that. So most of the evolutionary thinking ignored all of that stuff, which was at the genetic level. This was at a
Yeah.
pretty high level. Throughout this process, I was actually going in another direction, interested in the nervous system and how brains evolve.
Mhm.
That became my work for the next 2 decades. But in any case, trying to put that together. But the problem with brain evolution, of course, is, I mean, brain development in particular, brain evolution depends upon brain development, and all of the information that is building brains is mostly generated on the fly during development,
Mhm.
even though it's constrained and biased and shaped by the genetics. But the genetics that differs mouse brains and human brains is basically nil. But the amount of structural information that has to be generated, of course, is orders and orders of magnitude difference. And that has to be generated by an evolution-like and selection-like process. And so I realized that you couldn't talk about one without the other. That drove my work all into the 80s and 90s. And throughout the 90s, I was, again, still mostly doing evolution of the brain work and development of the brain work, doing fetal neural transplantation in the brain and stuff, just to try to answer some questions. And it wasn't until I began to reflect on that work that I realized that you could go back to these old arguments and
Mhm.
reframe them in terms of the kind of what today might be called self-organizing processes, but I think that's too simple. But in any case, I realized that you had to put those together. And so that this led me from there. I think the first thing that caught my attention was the besides brains and all the features about brains and so on, and their plasticity and how they respond to the environment, how developing brains have to adapt to body structures they find themselves growing into. I hit upon this article that was written in, I think, 94 by a couple of Japanese researchers that had identified and cloned the gene in the pathway for the synthesis of ascorbic acid. And they found it in rats and then used it as with a probe to probe other species and found that humans have a totally degraded version of it. Eventually, we now know that it's degraded by the loss of certain exons, but it's also a frame shift mutation that probably kicks it off, so that it's complete nonsense in some respects. But what I then realized is that there are a whole bunch of other adaptations that now that primates in particular had become, in a sense, dependent upon fruit for their source of vitamin C, ascorbic acid that everybody else produces endogenously, this shifted selection onto a whole bunch of other traits, including changes in taste, changes in vision, changes in the glucose transporter in blood, all these things. Because but basically was selection on one gene now becomes selection across the genome on anything that maintains this effect. That sort of set me thinking, and this led me to this other problem where I was working on birdsong. I
Mhm.
think in that paper I talk a little bit about this work,
Yeah.
which I realized that bringing this species of bird into captivity relaxed selection on sexual selection,
Mhm.
and so that breeding was done irrespective of song. But as a result, song didn't disappear. It just got more variable and more factors like the vitamin C story. More factors began to influence birdsong, including what they were hearing after hatching and so on. And that, of course, led me to go back and struggle with all this stuff to realize that the theme and variation logic of organisms was in fact a general phenomenon that had to be explained by a more abstract notion. And you had to go beyond this sort of selectionist argument to
Mhm.
do it. And by that I mean, even though Darwin sort of had a hint at this problem by the duplication issues, he
Mhm.
understood a little bit about redundancy, but it's just a tiny bit of his story.
Mhm. Yeah.
Um,
Yeah,
what
he does directly speak to redundancy as a mechanism of transition in part six of On the Origin. I mean, I think one of the things with Darwin is that I mean, when I read him, I think you're reading a great naturalist who has a flair for deriving, general principles, but you're not reading a systematic philosopher. So
right, right,
he has a huge number of observations, and he may briefly consider how they are consistent with his principles and maybe how they might, help to overcome some, chapter six is what, like difficulties with the theory or whatever.
right.
But he doesn't, he lays a lot of breadcrumbs there, but he doesn't really systematically explore them.
Right,
Yeah.
right, and that's what I love about it. I mean,
Yeah. Yeah.
he's ecumenical. He wants everything, anything
Yeah.
that'll be part of the story. He just throws them out.
Yeah.
That's why it's so rich.
Yeah.
Um, but in all of this, what occurred to me is that the selection story, um,
Mhm.
and Darwin is interested. Selection is a removal process. It's a
Mhm.
subtractive process,
Mhm.
and Baldwin is the first to probably really point that out. That there's all the criticisms that Darwin had after the origin
Mhm.
about the source of variations,
Mhm.
and there's innumerable cases where people are going, well, you can't have blended traits that can't be blending. There can't be increment. They can't be just incremental because again, you have a blending problem. This becomes a big deal for people like Weissman and Baldwin and Lloyd Morgan, and they try to get around it. But both of them, all of them point out that it's the problem of where variations come from. Well, then, of course, the gene discovery problem, at the turn of the century, and then you move into the neo-Darwinism. And the whole source of it, the whole source of variation is random error. Now, I don't think there's anything wrong with that because when we talk about gene duplication and degradation, we're
Mhm.
still talking about random error. It's
Mhm.
just that it's random error in a highly constrained context. And it's understanding the constraint effect that was missing. But
Yeah.
in all of this, what I realized is the Darwinian story, the way we've inherited it, actually
Mhm.
we inherited most of the average guy in the street inherited it from Herbert Spencer.
Sure.
You know,
Yeah. Yeah.
the the survival of the fittest is Herbert Spencer, not Darwin. But but basically, it's a subtractive story.
Mhm.
And we had sort of lived with the story of addition by error. People like Osborne and others who actually realized that was a problem and said we need to have a second story. I don't know what it is he says, but he gives it the name Aristogenesis or something like that. Um,
Mhm.
uh, and then Lloyd Morgan tries to do it and so on. And it's not until just a few people begin to ask the question, where do new genes come from?
Mhm.
that it begins to become a problem, and then finally you get Ono, who I think
Yeah.
brings it together and says,
Yeah.
no, new genes can come easily because now we've seen there's whole genome duplication, and we've seen that hemoglobin genes are duplicates of each other. So to some extent, it gets settled there, and I think that part of it gets answered at that point. But what I realized in hindsight, and I have to say this is only in the early 2000s, so it took me 20 years to get there, is that it's a Darwinian story with only one change.
Mhm.
And so gene duplication is, I use Darwin's, I think even in that paper, I recited Darwin's three principles in 1838. There's, offspring or grandchildren like grandparents, variation, there's variation of offspring. And then there's, I don't know how to describe it, but basically it's the over reproduction with respect to the means of support. Um,
Yeah. Overproduction of variations. Yeah.
yeah. What I realized is that the gene duplication story, the story that Ono originally originates, the first two are captured in other ways. The
Mhm.
first one, the inherited story, is duplication.
Mhm.
Inherited is just sometimes exact duplication, sometimes varied duplication. But the second part is the second law of thermodynamics. Second law of
Mhm. Yeah.
thermodynamics is you just leave things alone and they'll degrade after a while
Sure.
if you don't repair them. The third part is the part that's inverted because in gene duplication, with the exception of dosage problems, there's in effect there's not much selection going on.
Mhm.
And so one of the questions that becomes Ono's question, of course, is that how do you maintain these? And we get people coming up with answers to this, like all the way to the 90s, late 90s, that so long as duplications don't produce anything negative, there's a kind
Mhm.
of neutrality or partial neutrality to it. And that's the opposite of selection.
Mhm.
Basically, selection is that only a few varieties will survive because there's a limitation. If there's no limitation, you can multiply varieties. And if you multiply varieties and you allow the second law of thermodynamics to still be working, then those varieties will degrade a little bit. They won't degrade fundamentally. Some of them
Yeah.
will become pseudogenes, and particularly if they stay out of each other's way by different expression patterns and so on. In fact, the redundancy may not have dosage problems. And of course, you in your paper even talk about many of those.
Yeah.
Yeah. So what I realized is that it's really just inverse of the selection part. But if Darwinism is now just thought of as natural selection,
Yeah.
or
Exactly.
or
Yeah.
or now David Sloan Wilson who calls it multi-level selection.
Mhm.
But selection is always the negative.
Yeah. Selection
Yeah.
is always secondary. I mean, I think Darwin's first principle, really, if I put them in their sort of ordinate, their order, their ordinality is variation. Overproduction of variation is the first thing. And I mean, I think it does correspond with firstness, spontaneity in Peirce, but that's
Yeah,
a
yeah.
different discussion. But Peirce does map those onto each other, right, in a guess at the riddle. But yeah, so I think that's often kind of ignored. So I think you were right to say it's sort of our inheritance of Darwin in a way where that gets ignored. And I think it's also the attempt to mechanize Darwin's thought in a way, right? Because
I,
in the mechanical models of, say, statistical thermodynamics, you have a predefined state space, which is essentially, closed, essentially finite, right? Whereas in Darwin, he's not thinking in that way. He's not trying to formalize. So variation is just a given. And in some sense, because of its vagueness, it is an abstract principle. He doesn't have mechanisms for variation, which is the point you were just making. But we can take it as a kind of ongoing, inexhaustible, not fundamentally pre-statable or predefined. It's not a space as such, right? It's not.
right.
And so I think some of these difficulties arise not so much with Darwin's own writing, but with the attempts to formalize it, with the attempts to identify mechanisms, with the attempts to identify privileged bases of reduction, right? Like the gene, which again is a classic sort of effective theory type approach, right? So all of those attempts to make Darwin more mechanical, more rigorous than he actually is, are where I think some of these problems perhaps arise. And so when I hear the inverse Darwinism thing, I think, yeah, inverse Darwinism, but I don't
Yeah,
necessarily think it's really an inversion of Darwin per se, right? Of his own writings.
that's
Although I would,
exactly right.
yeah, I would
Yeah.
also that he does again because he's not really systematic. He contradicts himself quite a lot, and sometimes he does overemphasize the role of selection, or maybe the Malthusian intuition is sometimes overemphasized at certain points. And if we were to attempt, and I think that would I don't know how productive it would be, but to go through systematically all the arguments and all the observations made just in the origin, without even reading all the rest of his work, we would find there are a lot of internal tensions. But ultimately, like you said before, that's part of the fecundity, part of the productivity of his thinking. So, I mean, one of the things I've wanted to, actually, there's a funny analogy with Ono there in the kind of molecular evolution literature, because there are a lot of people writing subsequent to Ono, like force and other, with their subfunctionalization, like other mechanisms that people are identifying and specifying. And they're sort of saying, look, Ono didn't have this. All Ono has is this birth and death thing. He doesn't, it's all neo-functionalization as opposed to subfunctional, it's etc. But if you actually read Ono, it's a bit like Darwin again. It's like actually he sees all of that stuff.
And mentions it here and there, drops
Exactly.
hints.
Exactly. Yeah. So they're similar in that way. So, I mean, one of the things that I would hit upon there. So, yeah, I think, yes, selection is a subtractive process. I think that's right, but it's not a winnowing of a fundamental, state space or something like that. That's the
That's right.
mistake to make, right? Because again, that overproduction or that constant production of variations is just an operation. It's just a constant thing going on. It's not a countable set of states that could then be, winnowed. I mean, I wonder if, and this is an aside, a plant flag, I wonder if this is something Persk does get kind of wrong with his evolutionary realism, where he imagines at the end of time, the crystal, like the world of pure secondness, where everything is
Yeah.
finally settled. And I'm like, well, that can only be the case if there's a finite set of variations that is being determined in some sense, which I don't think is the right way to think about evolution. I mean, in his defense, of course, he says that it's real but not existent final state, right? Like it's
Yeah, right.
an ideal. But yeah, I think one of the things that you've really identified, which I completely agree with, is that this notion of relaxation of constraint is a really, really, it's like a fundamental level or a generic evolutionary process. And I think that that is understood in evolutionary biology or in certain areas of evolutionary biology. But again, biology is we often talk about biology as if it's a kind of monolithic field or whatever, but it's so heterodox, like the natural world itself, perhaps. But I've analogized, for example, things like in molecular biology, we have escape from adaptive conflict as a model, right? So that's precisely this, you change location or something happens in at the ecological level, perhaps, which changes the selection, the action of selection on a particular gene product, and that changes the mode and tempo of the evolution of that gene product. And I've analogized that at the organismal level. You have escape and radiate, right? So this is essentially the same notion, which is that an organism in a particular, ecological set of relations in which it has evolved. It's under a bunch of constraints. It's got predators. It's got, predator-prey dynamics that it's involved in, perhaps on both sides. But there are all of these, encrusted, accreted, co-evolutionary dynamics, which are kind of keeping it essentially stuck in some sense. What it's doing works and it can't deviate too much. But if it happens to migrate for some reason, if it happens to get isolated, there are all sorts of ways this can happen, right? Basically, what's happening at the abstract level, right, is that the constraint, mechanisms, the operation of constraint is changing, the constraint dynamics are changing, and then that organism may, this is a punctuated equilibrium type of model. It's a catastrophist model in a sense, right? Like a catastrophe just being an unprestatable disruption of that set of norms that are currently constraining the evolution of a given lineage. That's disrupted somehow, many ways that can happen. And then you get this punctuated thing, you get this sudden spurt in evolution. And we can talk about like invasive species in this regard. You take a cane toad from the tropical Americas and you bring it here to Australia, and all of a sudden, all of its natural predators, which are resistant to toad toxins, are not present. and it can both obviously overproduce, it can explode in population, but that changes the evolutionary dynamics. There are weird things going on with toads and their migration in Australia, where you get this weird selection. Really fast moving toads who happen to be moving west faster than the others end up differentially breeding with each other because they're leaving all the slow and lazy toads back in the east, and so you get all these strange effects of selection for being a bigger and faster hopper, essentially, right? So to keep outrunning front of toad population.
I never heard this story.
Yeah, well, again, the guys, Rick Shine and his team. Rick Shine's one of the world's most famous herpetologists. They've done a lot of work on cane toads. They had tried to propose this as, again, a kind of addition to, or something not in conflict with, but distinct from Darwinism. And again, I think, maybe, I mean, it's a novel mechanism, is what it is. But is it really distinct from, the kind of scope of Darwin's thought? That I'm less sure about. But yeah, what I love in your thinking as well is the complementation aspect, so the relational aspect to your thought, right? And also
That's the critical piece here.
yeah,
In the book I described this, inverse Darwin is the yin to Darwin's yang. Um,
yeah.
that
I mean,
you've got to have them both. You've got
yeah,
to have, and this is true for logical systems as well. You need introduction rules and elimination rules. You can't have a complete system without both that are in balance. And that's, of course, got to be true for the evolution story. It's just
yeah,
that neo-Darwinism had reduced the additional problem, the introduction rule, to just damage, just noise, and
mhm,
not recognize that it's always structured. The other thing that is crucial to this is that the first part of it, that is excess,
yeah,
producing excess. Life
yes,
has to produce excess to stay ahead of the second law of thermodynamics so that it
yeah,
will always produce excess. And excess will oftentimes relax selection,
yeah,
protect against error
yeah,
accumulation. And so the two of them together, the fact that life, this two sides of the second law of thermodynamics. One is to keep ahead of it, you have to produce excess. And the second one is if you have redundancy, it protects against error. Those two things automatically set you up. Now, if you have the third part, which is the limitation of resources on excess, or you have no limitation on excess or variation, that's the only difference between the Darwinian story. So what I was getting at is that this is a really one has to stand back that the Darwinian story actually does include this implicitly. Um, it's just that Darwin begins in 1838 in his notebook. He writes this stuff down just after finishing Malthus' book,
so
and
he's
so that he sees that answers a lot of his problems, but it sort of leaves us all with this new problem.
I mean, I
yeah,
think another big difference is that Darwin focused a lot on positive mutations, like the
mhm,
mutations that would be selected, whereas in reality, it's negative mutations that are neutralized that
mhm,
allow the space for complementarity. So I'd say even in his version of events, it's still the opposite products that are kind of
mhm,
integral to it.
One of the differences that I wanted to bring up between us, not that you don't think these terms, but in fact it's not as central to the toxin story,
mhm.
is that what I've been writing about is synergies.
Yeah. Oh, but
Um,
it's so central. We'll get to this. Yeah, that's where I was going to go. But yeah, please continue.
Yeah, so we don't yet, we have terms to talk about, neo-functionalization, sub-functionalization,
Yeah, yeah,
all this stuff, moonlighting, whatever,
yeah,
all
yeah.
these ways we have to talk about it. But in fact, I think the interesting stuff, I shouldn't say that, but one of the things that I'm interested in is a system that should, by virtue of the nature of competition and selection, should make synergies, cooperation, altruism, however you want to think about it, should make
Mhm,
it very unlikely to evolve.
mm,
Um, it's hard to select for it.
mm,
And so it's one of the things I like about Ono begins his book with a preface in which he uses the old phrase all work and no play. Um,
mhm,
that he wants to talk about the play side of things. So it's because that's what we're really talking about here is the play
mhm,
side. And play has to be in a context of protection, of
mhm,
error tolerance.
Free sporting, as
Yeah,
Peirce
yeah.
would say, right? Yeah, no, yeah, there's so much. So I think the on the excess side of things, like I think that's such a key concept. And there in my interactions with continental philosophy, I even turn to people like Georges Bataille, but Deleuze, Gilbert Simondon. There's really interesting stuff on excess there. And again, I think this is fundamentally a way that sort of the evolution of life and just the dynamics of living systems can't be reduced to sort of mechanical models because excess is in some sense primary. But I'd say, one of the forms of excess, so you identified two things. You said producing excess to stay ahead of the second law, and then you said the redundancy story. That's also excess, right? That's the production of excess variation, right? Which then is grist for the mill of evolution. So that's the story about the evolution of evolvability, which I think we can keep drilling down on that story. I mean, just on the synergy side of things, I mean, there's so much to say there, right? In the chemical ecology space. So one thing is toxins, right? And toxins are, so chemical ecology is the study of molecules that organisms use to mediate interactions with other molecules, right? And toxins are a particular mode of that, an antagonistic mode, right? So a toxin is a molecule intended to have a deleterious effect on another organism for reasons of predation, defense, parasitism, etc. And so, yeah, tons to talk about there. In the broader chemical ecology frame, synergy is the key story, right? It's fusion of lineages, right? So, like, ultimately, the endosymbiosis story, which you talk about, that's a kind of a limit case of a very ubiquitous dynamic in chemical ecology, where a lineage becomes dependent on the chemical products. And I don't mean in an energetic sense, right? I don't mean in a thermodynamic sense. This is why, again, plant a flag. We don't have to go into this, but we could. Why I kind of object to the notion of operational closure as it comes out of cybernetics and into autopoiesis and all of that. Like organismal lineages and chemical ecology is just like completely full of examples, are constitutively open to the operators of other lineages, which are either forcibly or voluntarily, various different ways, introduced into their own molecular economy and reorganize it, right? But exactly as in the ascorbic acid example, which I absolutely love that, because again, I do a lot of work on what I call human chemical ecology as well. So this is the human relationship with drugs, and so we can talk addiction and all that stuff too.
Right.
This is like in chemical ecology, I make a distinction between disruptive pharmacology and normative pharmacology. And one of the interesting things is the way things transition between those modes. So the way an exogenous molecule, which is disruptive in the target organism, can actually become part of what is required. This is like an addiction dynamic, right? What is required to maintain that, molecular economy. So that's another thing. But synergy is just so ubiquitous at that level. Like, for example, this is something I've brought up a number of times, but in marine ecosystems, a huge variety of invertebrates that are metamorphic, so that have multi-stage life cycles, and that have a transition, as many marine invertebrates do, from a planktonic larval phase to a benthic and sessile adult stage. They often cannot undergo that metamorphic transition without certain bacterial metabolites that they themselves obviously don't produce. So basically what happens is they're getting buffered around, they're moving around in currents, they come to an area with, rock, coral, whatever it is, they start contacting those surfaces. And when they contact the right kind of biofilm, they pick up enough of a certain metabolite that they can begin that metamorphic process. And without those metabolites or effector molecules, whatever you want to call them, in that context, they simply can't do it. So they can't complete a life cycle without an operator that they get from another lineage. And again, that sort of thing is really, really ubiquitous, and we could come up with many, many, many examples. It's kind of what chemical ecology studies, right? Because all of these co-evolutionary relationships that are studied in chemical ecology are essentially obligate on various levels, And again, I do think Darwin is hip to this in his interest in like pollinators and pollinators.
You're right.
Right. So co-evolution, again, there is an overemphasis. And in our reception of Darwin, but maybe it's there in Darwin himself, maybe he's responsible on competition. But cooperation, he's not exactly unaware that there's a great deal of cooperation, that, mutualism is not a phenomenon that would have, surprised Darwin, right? He does talk about it. Again, he's a great naturalist, so he's seen all of this stuff. And that is what has fueled his imagination to come up with these principles. But he never really, and one never does with biology, because every time you think of a rule, you find half a dozen exceptions to it. Right?
Right.
That's the thing about biology. But he never got through systematically integrating all of those things. And it's a good thing too, because we have we've had nearly 200 years of biology since then, and there's still plenty of work for the rest of us to do. But yeah, he sees a lot of those things. But also, just to introduce another thing, in that paper that you read, the How the Toxin Got Its Toxicity paper, there's one way of thinking about synergy, which is the origin of novel functions in the more or less arbitrary or stochastically mediated encounter with novel interaction partners at the molecular level, right? So it's the way that stochastic gene expression or, arbitrary mutation in regulatory sequences and that sort of stuff, as a result, sometimes of redundancy, results in molecules being expressed in places or at quantities which they haven't been selected for, and thereby encountering a novel range of interaction partners, whether that's just in another tissue of the organism that is already producing them, or it is even more dramatically, and this is the story with the origin of toxins, right, in the body of a target organism into which they must necessarily originally have been accidentally introduced. If you're a lizard that subdues your prey by biting and chewing on it, you're introducing a bunch of whatever's in your saliva into that target organism. And maybe you've got things in your saliva that have an antimicrobial function because you also do a bunch of scavenging and blah, blah, blah. But when they're introduced at a certain quantity, there are all sorts of variables here. What's the physiological state of the target organism at the time? All of this stuff. Unending weeds to get into. And again, why it's very difficult to come up with sort of one-size-fits-all models for these sorts of things. But it has an effect on weakening that organism, which gives you a 2 or 3% greater chance of subduing it in that tussle. And then we're off to the races with some sort of positive feedback mechanism on the selection for toxins. But again, what's key there is that molecule, which had some regulatory function or whatever in the body of the producing organism, encountered a bunch of other interaction partners or at different levels of concentration, etc., in the target organism. And that novel molecular ecology, so distinguishing, these terms are all already overdetermined. Chemical ecology, the study of organisms using molecules to mediate interactions. Molecular ecology is often the attempt to connect molecular biology with ecology, right? I'm talking about the ecology of molecules themselves.
Yes, right.
Right?
Within a body, yeah.
Yeah, exactly.
One of the things I thought of reading that paper was that we're really talking about synergy versus anti-synergy.
Sure.
That is, that the toxin story is the perfect complement to the synergy story. What's happening is that they're both involved relational systems, both involve a kind of ecological interaction, and yet one, because it involves two different organisms, has an anti-effect, and one, because it's happening within an organism or within a community, has a synergistic effect. But they're effectively, you might say, the left and right hand of the same problem. I have not acted because I don't know the toxicity part of the story.
Yeah.
I saw that as a sort of wonderful complement to the synergy part of the story.
Yeah.
And what I was thinking is that we don't have a good term for the synergy side of it, and we don't have a good term for the anti-synergy side of it. I don't have a term for it, but I'm thinking that making that distinction and making it clear is probably an important move. And it's certainly one that I want to take up since, by the way, the title for the book that I'm writing is called Falling Up. And it's a stolen title, actually. It was a title by one of my favorite children book authors. A guy named Shel Silverstein. But anyway, what I want to talk about is how complexity and synergy is the result of falling, of actually removing constraints. And so I just wanted to get that idea across by this kind of a odd juxtaposition, the oxymoronic nature of it. But
I like it. I said to Parham yesterday, I sent him a song by the band Black Eyed Peas, like from quite a long time ago, before they became a really big, if you know of them, they were quite successful, called Fallen Up, with an apostrophe, Fallen Up. Yeah.
Wow, really?
Anyway, it's just what he told me the title of your book, and I was like, oh, well, this is a great song.
All right. Well, send it to me also.
Okay, I will. Yeah. It may be of tenuous, the relation to what you're working on might be quite tenuous, but yeah, it's still fun.
So a lot of this was the result also of the work that I was doing in my book Incomplete Nature,
Yeah.
which is really an attempt to sort of bring information theory, semiotic theory, and biological theory together and to make sense of it. Interestingly enough, I started to write the book in part to sort of finish my work on the development and evolution of brains and what that tells us about how cognition works. Never got to the brain part.
Yeah.
But that's another piece of the story that I'm very much interested in recognizing that the generation of biological complexity has this logic that's very different than engineering logic. It's not a logic in which you're adding things, putting things together and seeing if they work together or not. It's actually a very different kind of process, and it mostly involves a kind of a differentiation, loss of specificity effects, and so on. Differentiation, then recombination effects. But what I realized is then studying how brains have developed. Of course, the development of brains in developing organisms has these evolutionary analogs, very obvious evolutionary analogs. And what I realized is that there is no point at which the brain loses that dynamic and starts to become a computational dynamic. The computational dynamic is the engineering dynamic. Brains function to produce complexity the way organisms produce complexity everywhere. That's a totally different dynamic. So that one of the reasons that I've gone in this direction is that I realized I had to understand that dynamic in an abstract sense before one could actually go back and say, no, we've really misunderstood how brains work. And this is one of the big problems that we're still facing and why we're confused by things like artificial intelligence and so on. Why we get confused and thinking that it's something that it's not. So in that respect, you were talking about how we in biology recognize that our rules always have more exceptions than we want. And yet there are general principles, general tendencies. And that's what we really need to focus on. That's pretty much what Darwin was focused on.
I totally agree. I think what just needs to be said there, which I think is very consistent with what you're saying and very consistent with Darwin, is that the more general your principle is, in a sense, the more vague it must be, right? So you're going to sacrifice specificity in order to achieve generality. So I always say, if someone asks me, why does a baby brown snake have different venom from an adult brown snake? This is one of my little discoveries during my PhD, that indeed they do. They have an ontogenetic shift in their venom composition. If I were to say natural selection or evolution, I wouldn't have explained anything, would I? It would be true. That is part of the explanation. But in a sense, that's just the minimal posit that I need to get traction to be able to then wade into the incredible specificity of that organismal lineage and what's going on with it in order to begin explaining why that might be. And then I think because variation is the first principle in some sense, I should always expect a kind of what Schelling would call an indivisible remainder, right? My explanation's never going to be complete, because there's always going to be some degree of arbitrariness that is just we can't reconstruct that evolutionary history, because there is just, on a particular given day, this just happened to, there's some eventual aspect to it. Like firstness, again, is operative there. And it is essentially, it grounds the possibility of explanation, but is not in itself explicable. Like, I think that's how Peirce understands variation, all right? Yeah, look, there's so many things, again, like the synergy versus anti-synergy thing. I really like that. And I think there's a very general principle here as well, which is, again, it's to your point about why evolution is not, amenable to engineering logic, is because it's not fundamentally a linear optimization process, right? In fact, I would say it's not quote unquote, again, fundamentally an optimization process at all. Optimization occurs, so adaptation occurs, but divergent, like that's a kind of convergence, right? Divergence is also always occurring, and different organisms will have, this is like an explore-exploit type trade-off, or a specialization versus generalization type trade-off. Different organisms may more heavily invest in convergence, and others may more heavily invest in strategies for divergence, So highly generalized organisms are, again, they are those ones that can probably accumulate a lot more neutral variation within themselves, which means that they have a kind of grist for the mill of a very context-specific selection, which I call elicitation, which is basically saying, like, this is what you need to be right now. You know, you can be all these different things, but right now you need to be the one who's running in that direction, or what, you know, there's that inherent plasticity of behavior, flexibility, which is grounded in this fundamental spontaneity, right? So organisms are nothing if not spontaneous. But yeah, I love this. Yes, synergy versus anti-synergy. It's something that I've thought about in terms of, like, again, the normative versus disruptive thing in pharmacology, but, you know, where if you were thinking in fitness landscape terms, you know, the fitness landscape of two lineages are kind of converging on each other and then merge in some sense, maybe in the endosymbiotic limit case, but also where they are fundamentally completely at odds with each other, right? And the term that we use for that, I suppose, in toxinology literature is a chemical arms race. So this is a kind of a red queen type thing in some sense, I suppose, but you know, the toxin target coevolution, it's often, you know, there's an attempt to evolve resistance and an attempt to overcome resistance. And really fascinating thing, like with any arms race, really fascinating things can happen here when, you know, innocent bystanders accidentally get involved in a long-standing arms race, say between a toxic organism and its target organism. So in Australia, you know, we have paralysis ticks, the Ixodes tick, Ixodes holocyclus. And they produce this horrible neurotoxin, holocyclotoxin. And what we believe basically is that their natural, quote unquote, natural, their normal host organisms are mostly marsupials. And indeed, if you find a wild marsupial, particularly in subtropical and tropical parts of the country, that they often have a number of these ticks, but they're fine. You know, they're just operating as normal. And indeed, it turns out they have very strong acquired immunity to this toxin. It's acquired, so it's not innate, it's not genetically mediated, it's antibody mediated, and they initially get it from their mother's milk, right? So it's passed from generation to generation, and they maintain it. But if you isolate them in captivity for a long time from ticks, they become susceptible to ticks in the same way that dogs and humans are. And dogs are killed all the time by these ticks. That neurotoxin. And so the neurotoxin is there because the tick, it's a soft tick, starts out really small and gets really big. It needs to feed for about 5 days, 5 or 6 days to achieve, you know, full satiation. Marsupials also have these incredible syndactyl claws. They've got fused digits, which we believe are grooming claws specifically for removing parasites like ticks. So for the tick, the main thing it needs to ensure it can feed for 4 or 5 days is to avoid detection. So it produces a neurotoxin, which is an analgesic, so that the marsupial doesn't feel that it's been bitten. But it has to produce so much of it just to have that intended analgesic effect on an animal with acquired immunity that it can kill a dog or indeed a human. We have antivenom. We have tick antivenom in Australia because of this. And then, you know, there are other examples like funnel web spiders, which are bizarrely toxic to primates for no real evolutionary reason. But again, you get this. I mean, it is an evolutionary reason, but it's not a selected reason, right? Not a functional reason. You get this with redundancy because the toxins that these funnel webs produce are in these massively redundant arrays. So they've got dozens of isomorphs, and they're all essentially freely varying. And some of them are, you know, just happen to be highly toxic to primates, even though there's no ancient history of exposure of those spiders to primates. So yeah, so this arms race thing, though, which I think is basically your anti-synergy notion, it is a really, really common dynamic. Again, I think what's really interesting is the way that in these sort of symbiotic relations, and it's easy to think about with the classic categories, parasitism, commensalism, and mutualism, organismal relations can shift across those boundaries dependent on very, very contingent facts about, say, the metabolic state or the just, you know, general health of, for example, the host organism. Like aspects of your microbiome can go from being mutualist to being, you know, or commensal perhaps, to being parasitic if you just get run down. You know, if you're not getting enough sleep, something that is otherwise existing there in at least a neutral way can start to make you sick. And I think this is something that's happening with these organismal lineages all the time. Again, with endosymbiosis as this kind of big ticket example, what might have been an antagonistic relationship, either parasitism or you know, just predation or whatever, has become this mutualist And now it's not even mutualism, right? Now the lineages are completely fused. But I think that, again, that kind of thing is happening all the time. So that synergy and anti-synergy thing, those are the two poles of a spectrum, but relationships can shift across and transition from one pole to another.
So in fact, this is something that the two of us have been talking about in terms of the oxygen poisoning event in evolution.
Totally.
That in effect, what becomes a protection to oxygen, taking in some bacteria that protect you from oxygen, you can now begin to use the energy that they're able to protect you from. And we've been talking about this in terms of very early life story. I won't go into it because it's pretty complicated, but basically that how was it that phosphates first got recruited? Were they initially like the oxygen story? Was there necessarily some kind of protection from phosphates? Because of course phosphates are highly energetic and break up damaged proteins and so on. And was the acquisition of phosphate capacities by virtue of ATP and related molecules, by nucleotides and nucleosides, was that originally a protection feature that then became recruited as an energy feature? That in effect, that logic, that shift from protection to taking advantage seems to be a general feature that I think fits this argument very, very well. This shift from anti-synergy to synergy, from defense to taking advantage. It seems to me we need to look more at that as a way to talk about this. And when you're just talking about the ticks, I was wondering if there isn't a toxin antitoxin story to be told there. You know what I'm talking about? The toxin antitoxin story, which says basically that if you get rid of one, the other becomes a problem. So you basically have to maintain both.
Yeah.
I'm wondering if there isn't something like that that's probably going on, that in effect the marsupials that are producing the antitoxin effectively, are they disadvantaged when they don't carry a tick load? Is there actually a disadvantage to producing this antitoxin?
Well, like autoimmunity, for example,
Yeah.
and the theory about our loss of nematode parasites leading to our sort of immune vulnerability. Yeah, I really like that idea. I mean, it's a certain sort of like, whatever doesn't kill us makes us stronger kind of notion, right?
Right.
Yeah, that sort of stress leading to a degree of robustness. I mean, it makes me think a lot about human drug relations as well, of course. And again, how quickly something can go from being essentially toxin-like, but certainly disruptive, to becoming, normative, becoming a necessary constitutive part. But then how in something like delirium tremens, when you remove that alcohol, that all of the things that have been upregulated, your gene expression has changed dramatically to create this novel molecular economy, which is coping with the massive influx of these toxins. Now you're going to go into a state of autotoxicity if you remove the that exogenous toxin. And I think these things they're so common and they happen so quickly. Like one of the things I like to talk about is the glutamate rebound. So this is just you don't need to be an alcoholic or anything. This is a single night of drinking, right? And once you drink a certain amount of alcohol, so you have to drink a fair bit, you start to get glutamate suppression. And if that goes to a certain point, you pass out, right? I'm sure none of this has ever happened to any of us in this conversation, but I hear these things happen. But what happens then when you wake up, or when this hypothetical undergraduate student wakes up at 2 or 3 in the morning, having passed out, and switches on the light, is that the lights are incredibly bright. They hurt their eyes. They've got this, it's obviously an incipient hangover, but they have a particular sensitivity to light and sound and all of that. That's glutamate rebound. Now you're in a state of glutamate toxicity because you went through a state of glutamate suppression, and then you had an upregulation of glutamate receptors in response to that. And so then when your glutamate production returned to normal after the alcohol was all processed, all of a sudden things are going wild. You've got too much of your, primary stimulatory neurotransmitter, and now you're like wired, And so what's fascinating about that kind of thing, I mean, it's fun to talk about. I like to talk to my, pharmacology students about this kind of stuff, but it's also how quickly it happens, right? So this is a kind of experience-dependent plasticity that's happening at the level of gene expression, but it's happening so rapidly. And again, if we think about that kind of thing in chemical ecology more broadly, these sorts of things are happening all the time in organisms. They are incredibly dynamical systems, obviously. And some of these things are potentially turning into budding off of novel lineages, or at least novel directions of evolution at the level of lineages.
And also, it's the example of the addiction and recovery and the that transitional period. We're also talking about different time domains. That
Yes.
if the alcohol toxicity very rapidly damages something, the
Yeah.
system compensates for it, but the compensation was one of gene expression, and that's
Yeah.
going to be slow to change. So
Mhm.
this is true for opioid addiction as well. What's
Mhm.
happening is you shut down certain things because these neurotransmitters are saying, it's got too much of this neurotransmitter. I got to decrease my sensitivity. Yeah, that's a metabolic effect, and the metabolic effect will take days to
Yeah.
recover. So
Yeah.
there's a
Yeah.
long-term effect.
Yeah.
In the 90s, I was doing a lot of work on Parkinson's disease and the use of L-DOPA as a response.
Mhm.
What's going on is L-DOPA is replacing some of the dopamine function. But one of the things that's also happening is the receptor
Mhm.
synapses are becoming more and more sensitive. So that at some level, what happens is they're oversensitive, and all dopamine does is cause you to have tics.
Yeah. Yeah, I mean, this would be a whole conversation about withdrawal and
Yeah,
and all,
exactly.
there are all sorts of things going on here. I love that you introduce different timescales, and I mean we can even put withdrawal and addiction as similar mechanisms on different timescales in
Yeah.
the sort of pharmacological world or human relationship with drugs world. But I wanted to go back to something we could talk about more, which is what you've said a couple of times about brain development and the way that it is analogous to an evolutionary process. And of course, Edelman and others have sort of made this point, right? But I think it's all developmental processes, I think is the point you're making, are analogous to evolutionary processes. And so I wonder if you've had any contact. I think there are a number of people who've said this in different ways, but I wonder if you've had any contact with the work of Jean-Jacques Cupiec, the French
I have
um,
not. I have
biologist and
not.
professor of biology. Yeah, he's got a book called, I think it's The Origin of Individuals. And he coins this term, I think he coined it ontophylogenesis. So he's talking about, again, the general, sort of at the diagrammatic level, set of general principles that apply at these different times. I mean, so we could talk about, it being scale free or fractal or whatever. And of course, a lot of, systems biology and sort of complexity theory makes that kind of move, free energy principle people making that kind of move, etc. But yeah, Kupiec is really interesting, and his ontophylogenesis is really interesting, partly because he's a very lucid writer on Darwin, actually. And he's a very lucid writer about, say, what Schrödinger gets wrong about biological systems. But really, it's just this notion that, again, for Darwin, the first principle is disorder or is variation, and everything has to, and that's why he needs a principle of selection to explain why variation, the distribution of variations isn't completely random or continuous. There are clumps in variation. That's what the principle of selection is there to explain. But again, because it takes for granted or takes as given that this variational first principle. Yeah, Kupiec is quite interesting. He edited a journal issue in 2012 called "Chance at the Heart of the Cell," which is very interesting. I think there's some stuff in there you might want to check out. But yeah, a big person for popularizing the idea that, again, not original to him, but that stochastic gene expression, for example, and also the non-stereospecificity of molecular interactions is both a refutation of neo-Darwinism, but is more Darwinian than neo-Darwinism because these are variational processes. And so, yeah, those are grist for the mill of evolutionary novelty. And again, I didn't get this from Kupiec, but that's very much the way that I had always been thinking about things in terms of the evolution of novelty in the systems that I'd studied, toxin systems, chemical ecological systems. It necessarily must be the case that molecules are promiscuous, they're sticky, they can stick to things that they hadn't got any selected history of interaction with. And that always opens up the possibility of an incipient function, or something that can be exapted, a propensity for interaction that can then be exapted and turned into a function via amplification.
So if you would share that name with me at some point in time.
Yep, yep, absolutely.
So I can look it up. Interestingly enough, it was my 97 book, Symbolic Species, in which I lay that argument out in great detail. I was arguing that you couldn't understand how human brains evolved and why they're different than other primate brains without understanding their development. And the other piece of that led me in this direction that we haven't talked about is Baldwin and Waddington. And first of all, how they're confused and shouldn't be confused, and how they were dealing with this problem, trying to resolve this problem. And I think both, in one sense, missed the target, but were on the right track. And so I wrote a number of pieces in the late 90s that were trying to figure out how to resolve that, because I used it in my book in 97 to talk about how language evolved. That language, in a sense, I was arguing it was a little like what Baldwin was suggesting, but it couldn't quite be what Baldwin was suggesting. And the main reason for that is that Baldwin was basically talking about how relaxation and selection, though he doesn't talk about it in those terms. This was a relaxation and selection argument. And what happens with relaxation and selection is that you don't generate strong selection in consequence. But he was sort of suggesting that it allowed new selection to show up. And what I was realizing that in fact, you relax selection and things tend to vary more. Variations accumulate, just the opposite of what he suggested. And then you get people all through the 70s, 80s, and 90s confusing Baldwin with Waddington. And Waddington, in a sense, is actually talking about what happens when you change selection. So you raise fruit flies in heat. What happens? So you've actually exposed developmental features that were basically hidden previously. So it's much more like what you've been talking about in terms of the ecology, shifting things into a new ecology and what happens. Totally different arguments, but people were confusing them. I would say that probably my first move in this direction was actually trying to clear up that problem.
Yeah, that's really fascinating. And I obviously have a great deal of your work to read, Terry.
Similarly, what we should do is also I want to get more of your work.
Sure.
So I get a sense of it because I'm going to take some of your arguments and add them to a couple of chapters in the book, because I've said no, I talked about the toxicity, anti-toxicity story, but actually this evolution of toxin stuff is perfect for all the reasons that we've talked about. It's perfect because it's the ideal complement to the synergy story.
Absolutely. One of the things I wanted to connect to what you were saying about language, and I'm trying to look in my notes to your degeneracy paper. One thing we could talk about actually is the relationship between redundancy and degeneracy, as those things are defined in the kind of molecular biology literature, and there's a kind of stipulative or almost arbitrary difference there. But degeneracy, unlike the way you're using degeneration, it doesn't necessarily mean like a degenerative process. It's like degenerative ground states of a spin glass or whatever. It's like multiple different ways of achieving the same end. And of course, the way they differentiate it from redundancy is that when they talk of redundancy, it's supposed to be, two copies of the same thing that fulfill the same function, and degeneracy is two different things. And obviously, at the ends of the spectrum, that's a meaningful difference. But I always have the question of like, where are we going to make that cut? So when I've got lots of members of the same gene family, are they redundant or are they degenerate? Because I can tell you they're not the same. They're going to have some point mutations that are going to differ. But jumping from that, the discussion of language, and I couldn't find the exact way of making the point that I wanted to make, but it's just an idea that I'll throw out there, and I'm sure you'd have something to say about. Because you're talking about the use of you're talking in general about relaxation of constraints leading to a certain degeneracy, and that opening up novel evolutionary pathways. And one of the things we also see a lot in toxin systems, and specifically we've seen it quite a bit with the evolution of venom in reptiles, is a very rapid kind of use it or lose it dynamic, right? So we see with snakes, for example, that even snakes with highly venomous, close, recent ancestors, if they switch to a defenseless prey, as we say, so they start eating eggs, for example, like fish eggs for some sea snakes and things like that, their venom systems very rapidly degrade. So, and we think that what's going on in these venom systems is already a very elevated level of evolutionary change, partly because of the redundancy that we're talking about, because you often have these, multiple gene copies in these redundant arrays, but you also have a kind of quasi-redundant system altogether. The venom system is required to some degree for securing a prey item. But it's kind of one of the things that you can't do without a heart, but you can do without a venom system if you're a snake, because you might be able to compensate, by switching to defenseless prey, or maybe by regressing to a constriction strategy or something like that. But also you don't have the same kind of gene dosage restrictions. In fact, you're in a situation now where more might be better, like if you want to produce more toxin. So that's another way that, duplicate genes can accumulate and you get more redundancy. But yeah, interestingly, we see it's just analogous to what you're saying about, relaxation of selection on masticatory adaptations. When we have stone tools, fire, I assume, would play a role there as well. Yeah, so that just another thing that I think is probably more common. Again, I think Darwin was aware of this, like vestigiality is something that he's aware of, but that disadaptation or de-differentiation can occur fairly rapidly in evolutionary time, and of course developmental time, right? Use it or lose it, being
and can be used, obviously accepted.
Yes, yeah, exactly.
And Darwin, I think, even recognizes this. I love his discussion of barnacles, and the degeneracy of the crustacean body type in barnacles. He gets that. He gets the idea, and he says, and how it can be repurposed, and yet it doesn't become part of his broader story. But it's again, like you were saying, it's one of his. How do you deal with the exceptional cases?
Yeah, and I mean, cave-dwelling organisms, all sorts of in that regard. Yeah. Another thinker, I'm not sure if you have had any engagement with, that I think would be of real interest to you, and I think there's a lot of convergence even in your treatment of information, which we could also get to soon, depending on how much time you have left, is Gilbert Simondon, the French philosopher, who's very, very, unlike, there's this generation of French philosophers, like just before the much more famous post-structuralists, who were very influential on those guys. There may have even been two generations, depending on how you count the generations, but that were very, very engaged with the science of their time. So, Georges Canguilhem would be one of the most famous, and Bachelard, Gaston Bachelard, and Simondon is another one. And I think that there's a lot of resonance. So Simondon's theory of information, he's coming out of a critique of cybernetics as well, and the insufficiency of the Shannonian and cybernetic paradigms for accounting for the way that information works in, say, living systems, right? So I think, again, you'd have resonance there. Do you want to talk about information, or do you want to talk about abduction? Because I'm quite excited to talk about abduction in the molecular context as well.
So the simple story of abduction, we started with information. I'll just say a few things. I should probably just send you this paper, and then we can have the conversation.
That would be great.
The part of it is to actually look at the breadth of abductive processes. So I start with Peirce, but then talk about Arthur Kessler's Act of Creation. I don't know if I think it's a very important book.
I haven't read it, but I've heard of it. So
you should pick it up. Even though it doesn't overlap with much of what we talked about. It's just a very insightful book. Then Gregory Bateson's idea of double description comes into this, and we use that one as an example. And then we use this idea of conceptual blending, all of which are sort of modern variants on the theme of abduction. And Bateson, for example, uses the example of binocular vision. And he says binocular vision uses redundancy and variation off of the redundancy. And as a result, by taking the systematic variation of the information from two eyes, that the difference actually this systematic difference creates a dimension of those differences so that we actually create a third dimension.
I just mentioned Simondon, so he's got this whole theory of disparation, which comes from exactly that analysis in the 50s now, binocular vision, and the need to solve that difference, that tension, by the creation of the third dimension, right? But so the integration in a higher order, like a complexification, essentially. That's the fundamental dynamic for Simondon's theory of what he calls ontogenesis. Very, very consistent. Yeah. Anyway, continue.
No, I should take a look at I have a close friend, one of my former students actually, who's totally into Simondon and tried to get me to read it, and I haven't spent the time to do it. So I probably should.
Yeah, it's worthwhile. Yeah.
Yeah, so that's interesting. But in any case, what I realized, I have a fairly simple example that I've used to start that paper, and I actually discovered this back in. I should tell you that my first paper on Charles Peirce I wrote in 1976, and it was basically it was called Semiotics and Cybernetics, the
Okay.
relevance of C.S. Peirce.
Wow. Okay. Sounds interesting. Yep.
And so it's what got me into Harvard actually as a graduate student. Somebody read it and said, "We got to get this guy here," but then they didn't treat me well, and so I moved on. But the key that I realized then is that there was a very close parallelism between his semiotic hierarchy and his inferential hierarchy. So that his inferential hierarchy, his abduction, was basically reasoning by iconism and reasoning by basically degeneracy of iconism. And so the simplest way I started out in that paper was to recognize that you can break down his way of trichotomizing inference by using just the simple syllogistic kind of an argument. So that in effect, using just three sentences. So all men are mortal, Socrates is mortal, and then Socrates is a man. That's your deductive version of it. The inductive version of it is that Socrates is a man, Socrates is mortal, therefore it's possible that all men are mortal. It would be nice to make it stronger by saying, well, Plato is a man and Plato is mortal. Now I've got two. That's the induction. Induction says you multiply cases in which the links of an individual to a property are the same. So basically what's happening is, again, the iteration is involving something like duplication. It's iterating, making probability stronger. But you take those same three sentences and you change the antecedent consequent relationship. So all men are mortal. Socrates is mortal. Socrates could be man, but Socrates could also be my bird.
Yeah.
Okay. How do you make that one stronger? Well, you say, well, Socrates is a man, and Socrates, no, excuse me, Socrates is mortal, and Socrates walks on two legs. Now it seems like I could say, well, then that might lead me to think that Socrates is also a man. But my bird is also that way, has those qualities. But as I iterate properties, walk on two legs, is mortal, can speak. Now, only my parrot will fall into this category. But if I add doesn't have feathers, then only my parrot that is plucked off all of his feathers will fall into that category. My abduction is still never fully accomplished. But by iterating properties, it becomes stronger. By iterating instances, my induction becomes stronger. By iterating, what I'm going to say is iterating, not properties, but so for example, all men are mortal and all mortals are born. That means that all men are also born. So in this case, what happened, and this is Euclid, the original, if you can get an original inference, then all those that fall within it, that can be contained within it, fit. But basically, taking just those three sentences, you can create the three forms of inferences. But now here's what's interesting about it. They each have to do with canceling the redundant elements. So all men are mortal. Socrates is a man. Therefore, I can conclude that Socrates is mortal because in the first two sentences, there's two versions of man. All men are mortal. Socrates is a man. I can cancel that and I can jump to, therefore, all men are mortal. In the case of abduction, the same thing is true, but you're canceling something different. All men are mortal. Socrates is mortal. Cancel the mortal, and I've linked Socrates with man. But in fact, you have to see the relationship. In each case, you're canceling the redundant element, and it allows you to see a new connection between two other elements. And it turns out that logic is very systematic, that you can see that it plays out, that the inference is the result of using iconicity and canceling the iconicity to get indexicality, to get one thing pointing to something else. And so this sort of drove all of Peirce's thinking. And so that in effect, what I realized is that iconic interpretation, producing interpretants that, according to Peirce, that recognize iconism, is about the same thing, is about, again, canceling similarities, ignoring differences. So that whole paper was written by basically saying that although Peirce didn't actually produce these things in this order, that the history of his ideas leads from his original thinking about inference to a more complex theory of not inference, but of semiosis, of actually representation. And that over the course of his life, he builds that. The only problem is that he doesn't talk about the competence to interpret. That is, what kind of a process does interpret? He talks about the process of producing an interpreter, producing another sign, but actually doesn't talk about the competence. When I was interested in what is different about human brains that makes us capable of interpreting the symbolic kind of communication that language utilizes, I was talking about a competence problem. That human brains allow us to produce something, Peirce's notion of interpretant, produce a kind of sign-sign relationship that made it possible to interpret things that don't carry any grounding for their reference. But Peirce didn't want to talk about any of that. He left that out of his work. And so he only produces a taxonomy. And unfortunately, the taxonomy became the driving force in all of subsequent uses of his ideas. People they love to use the terms and find an example of this, an example of that, an example of this, but as a result, ignore the interpretive process. And of course, that's what biology is. Biology is an interpretive process. And that's the link between them. And what I realized is that to some extent, what I've called inverse Darwinism, this generative process that requires both excess and the second law of thermodynamics, the degeneracy effect, that's in effect an abductive effect. Whereas natural selection is redundancy in the way that induction is, case after case after case of correlated functions. And some of them are regularly maintained and regularly not.
So will we say that when we were talking about convergence and divergence earlier, that when you say natural selection just there, you are talking about a convergent dynamic as opposed to the divergence, which we also are saying is taken for granted by what would become the theory of natural selection, essentially, right?
And so Peirce, you couldn't do any form of inference without first doing an abduction.
Yeah, right.
So it has the same structure. What I realized is that the success of natural selection is to produce something like a deductive relationship in which something follows by law almost inevitably. When you have a system and you've culled out all the variation that you can to streamline it, then it's like a deduction. That is, the rule follows not just probabilistically, but almost inevitably.
Until the context changes, of course.
So one of the key stories here is that, of course, you've cycled through these hierarchies. So once your function begins to fail, new abductions are needed.
Yes, exactly.
I've called it 1, 2, 3 repeat, that you have to do this again and again and again, both in producing the hierarchy of complexity in organisms, but in the cycle of evolution. So Matt, the guy who was sitting next to me here, has said we should call it an adduction. That is, it's this higher order or meta cycle through the three modes of inference. You
Yeah,
cycle through it, and then you, in effect, test it by induction, but you never get the perfect result. And so eventually, when you've converged things to a very simple, almost mechanical-like form, you now have the perfect context in which change context and it fails. And you have to go back to re-abduct it, so to speak.
yeah,
So
yeah, yeah.
so in this paper that I'll send you, we basically walk through this parallelism. And the argument, of course, is my attempt in the big picture to show that information theory is a subset of semiotic theory, is a subset of this broader theory that we think of as evolution, but it's larger than our evolutionary story. But it's all a similar kind of inferential argument.
Mm.
So that's really my intention in all of this, is to bring this down, and then of course we can begin to talk about cognition in the same terms.
Yeah, that's extremely lucid, Terry, and again, very resonant with, but different from. I'm coming from a slightly different approach, probably, to the way I've been thinking about these things. I mean, so one of the things that I think, though, is that is very similar. You know, abduction I think of as the genesis of a novel lineage in some sense, right? Which can that's a lineage of deduction, then, if you want, or induction, but that's itself happening at all these different scales all the time, right? So I'm not necessarily saying a novel genetic lineage or a novel species, but a novel, it's like a novel telos in a sense, right?
It's a hypothesis about
Yeah,
telos.
yes, no, for sure. I mean, literally for Peirce, right? So, and this point again about, you know, deduction works, so that lineage is viable until, or that strategy, let's say, is viable until the landscape changes. I mean, this is again why evolution is not a linear optimization process, is not describable as pure gradient descent either, right? Because we have to take into account the unprestatability, that's Kaufman's term, but the kind of non-fixity of the landscape itself. The landscape is always changing underneath you, or, you know, you're either climbing or you're descending,
Yeah, yeah,
depending on the metaphor. But, you know, the peaks and troughs are changing. And so as an organism, you have to, and this would be a Simondonian point, you have to, what he would call, maintain the charge of the pre-individual, which is that you have to maintain excessive variation such that you can essentially be more than one thing. You can be something else, which means you can continue to evolve, you can continue to adapt, or in Simondonian terms, you can individuate again, or you can generate a novel mode of being, which is what he thinks of as ontogenesis. So the way I think about this in its relation to the molecular redundancy story, well, I think it's really just all the things we've been talking about, which is that I think about, you know, molecular redundancy as something that's contributing to an inherent spontaneity that organisms have. So again, it's this excess of variation that is not, I would say, pre-canalized, right? It's not going in any particular direction. It's not for any particular like proximal function, but it is grist for the mill of the genesis of novel functions or novel directions. So just in the way that, you know, stochastic gene expression is kind of always palpating, in a sense, you know, what halos the organism or what is around. It's a way of kind of reaching out and touching the environment all the time, just as you could be doing that actually physically with a kind of spontaneous, I mean, I'm gesticulating all the time. I always use this as an example because I'm a big gesticulator. It's like, there's no reason I'm doing this. But, you know, if you're in a dark room, you start palpating your environment, you start touching the, you know, trying to find a wall. And then as soon as you get a response, right, you elicit a response. So I call this elicitation, and it's reciprocal, right? You elicit a response from your environment, which elicits a response from you. This is, for me, informative, right? This is informational dynamic now.
yeah, right.
There's a relational thing going on, which is a way that you orient or coordinate yourself. So you've essentially got free variation, spontaneous variation, and then something, like you can call it a signal, tells you which which subset of that spontaneous variation that you have operative in the background is the relevant one for you to harness at this particular moment. So in some sense, arbitrary motion and then the genesis of a novel direction. And this is just how I think evolution works in general, right? I think this is how the arbitrary and the emergent directionality of evolution works. And I think of that as abduction, right? That's how I've been thinking about it. It's kind of like, spitballing in some sense. This is hypothesizing, right? Throwing something at the wall until something sticks, but always in this context appropriate manner, always reaching out and touching and looking for a constraint, looking for a signal, which then to me, that's selection. That's what selection is, right? Selection is, it's saying, and this is the negative aspect of it, sure, you've got all this variation that's going on, but not that one, this one.
Right.
And that would the correlate to what you're talking about in terms of the cancellation of redundancy, right? That's a principle of selection, right? It's saying that this is the relevant variable right now.
Exactly.
You don't need all those other ones. We'll pursue what is laid ahead, what is gestured towards or whatever, indicated by this particular variable.
Well, and to some extent, what we're talking about is, in a sense, the hypothesis generation that has to take place at all levels. And
Yep.
Pearson's whole point of abduction was to say that hypothesis generation has structure. It's
Yeah.
not fiat. It's
Mhm.
not just fiat, whether it's Kekule's dream or Einstein thinking about general relativity by thinking about elevators versus gravity.
Yeah.
It's not just casting randomly. It has
Yeah.
structure to it. And the
Yeah.
structure is iconic structure.
Mhm.
And so basically, hypothesis generation is always based upon analogy, and which basically is a variant of duplication at the level of genes. They're analogous functions, but if you have the space to allow analogy, the space to play, then you have also possible interaction relationships that were never there before,
Yeah.
that can be explored without cost.
Is that where the structure comes from, though, right? So the structure is the context dependency of a particular relation. So I think there is the free sporting aspect, right? Of course, we could talk if any given empirical manifestation of variation is going to be highly structured, right? So gene expression may be fundamentally stochastic, but what's being expressed is not exactly random, right? It's still
It's constrained stochastic.
yeah, yeah, exactly. So there are adjacency relations, in a very abstract sense that we could illustrate for any given variational process. But what gives the hypothesis its purchase, so and that its directionality is something context dependent, right? So I mean, for Kekule or Einstein or whatever, that you could talk ad infinitum about the context. It's all the prior learning that they have for one, but it's their ambition to come up with novel hypotheses itself, right? It's their motivation. It's all of that sort of stuff. So it's not that just anybody, let's say, could have had those insights, but that there is, to your point, and I think it's to Percy's point, right? There's that psychic, firstness, whatever, free sporting aspect, which is ineliminable from the process. It is not a deductive process, basically, is all we're saying when we say that, right?
And I think what I want to be clear on is that it's not an inductive process either.
Yeah, sure. Yes.
The inductive
Yeah.
process becomes the testing.
Yeah. Yes. Well, it presumes that the goal is already set there. It presumes the directionality already exists.
Right. That's right. So another way to think about this is to compare it to the Lamarckian logic. The Lamarckian logic says that there's telos ahead of time that's driving it, as opposed to the emergence of novel telos.
Yeah.
So I think of the gene duplication story as saying that no, there was already a function. It was already integrated into the system, and we've duplicated it. You're coming from something that already works, so to speak, and playing with it. And you're playing with it in a context of redundancy, of, in a sense, error tolerance, of function loss tolerance. And under those circumstances, other things are possible. The Lamarckian story, in a sense, puts what you might call the seeking part of it out front.
Yes. Yeah.
Whereas this is a view that accomplishes the same thing without putting the seeking out front.
Yeah.
Basically says, no, there already was a function. This is, it reminds me of Pasteur's notion of the prepared mind. In each of these cases of Kekulé or Einstein,
Yeah. Yeah. Yeah.
Pasteur, it was because of their prior knowledge that they were able to go
Yeah.
here. But
Yeah.
of course, they were building off of their prior knowledge. They
Yeah.
were these were analogies based upon what they were already playing with. So
Yeah.
Pasteur is initially worried about testing spontaneous generation,
Yeah.
and worried about how it is that the wine barrels have gone bad, and trying to figure out what's correlated with it and so on. He's got all these examples there already, but he can now apply it to a whole new realm.
There is an aspect, yeah, I completely agree with all of that. There is an aspect, though, which again is very much part of what you're saying, but I think needs to be emphasized, which is the freedom from prior knowledge as well. So there's all this prior knowledge.
Yes.
Redundancy, it means that the constraint of that prior knowledge is not complete. So you are again, it's not unrelated to it, but you are deviating from it in a free manner. So there's that degree of stochasticity, whereas maybe it's counterintuitive for some, but if you put the telos, at the base, you imagine that seeking to some goal is already there. That is a kind of tyranny of the a priori in a sense, which means that there would be no possibility for arbitrariness, or for, this sort of quote unquote stochastic deviation, for something eventual, in fact, to happen only because of a unique confluence of things, because the thing is determined all the way back. I wonder though, Terrence, I don't know if you will of course read all of the stuff, but I don't know if you've written much about Peirce's evolutionary metaphysics. So I'm interested.
A little bit.
Yeah.
Yeah, and his three
Categories.
conceptions of evolution.
Yeah.
So very, very, and of course, very characteristically associated with what we were talking about, the three forms of inference.
Yes. Yeah.
They have the same kind of structure. And clearly, his agapic evolution is very much of what we're talking about to some extent.
Yeah. Yeah. Although I feel there's an ambiguity here, which is that we're also talking about Tychism, right? I mean, and of course the categories are all mutually, implicative and all of that. But with agapic evolution, it feels like there is a reaching, there is a kind of moving towards, which is always already present in that mode of evolution. And so I think, are we not talking about the way in which this infusion of Tychism creates novel agapic resonances or whatever, right?
But again, I think this is why the term constraint is so appropriate.
Sure.
In
Yeah.
engineering,
Yeah.
a constraint tells you how far precision has to be carried.
Mhm.
Within that, below that, there's a degree of tolerance.
Yeah. Yeah.
Below that, there's a degree of that.
Mhm.
So long as I've specified it this far, I don't have to do anything else. It can vary now
Yeah.
below that level.
Yeah. Yeah. Yeah.
And so the story is that it is always at some level of constraint, some level of tolerance.
Mhm.
And it's at the level of tolerance that, of course, or what I've been calling error tolerance,
Mhm.
that just basically says that no, it's just not fully constrained. The engineering model is typically we constrain it as far as we possibly can. But in
Yeah.
building a clock, even, the constraints don't have to be precise. They just have to be, the constraints of the material have to be such that they don't degrade. You can't use paper so much to do it. You got
Mhm.
to use something like metal. Wood will degrade faster, so you don't want to use wood. So there's a
Mhm.
bunch of tolerances you have to set up, but there's
Yeah.
no clock that will stay a clock forever.
Yeah. 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 But there's a multiple realizability of the clock function, the clock diagram, right? Yeah. Yeah. And I mean, this is exactly, of course, where the redundancy story fits so beautifully. The capacity to fulfill whatever your obligatory functions are whilst maintaining a certain degree of free play, for sure. I mean, I've been very interested in the kind of, and I know, Peirce tried this and sort of, scared himself out of it to some degree, this sort of firstness first kind of metaphysics, where he was trying to understand the origin of the cosmos from the generative nothing, and then he was told by someone, a critic, oh well, you've already presumed the capacity for thirdness to emerge and blah blah blah, and he sort of gave up on that. And I mean, he takes a distinctly theological turn in his late life, doesn't he? So it may just be a fool's errand to some extent to try and think like absolute origins or cosmogenesis or that sort of thing. But I'm very interested in a kind of speculative mode of thought, which is it's very, very consistent with all the stuff that we've been talking about, but which is really putting this variation in this position of firstness, which I suppose just means, I suppose in a sense, it's just what we're saying, which is that it is not completely constrained, and thus, and I just feel like in some sense this is what is. And I think this is the point that you've been making as well, is what's missing, again, not from Darwin's thought. I actually think a big part of what his innovation is in comparison to a lot of the evolutionary thinking of the 19th century and the late 18th is to really give this variational process its kind of ordinal priority, is to not try to always already have it subjugated to some, grand synthesis, some whole that's at the base of things, to really say, no, there is this genuine free play happening here. But I think, to your point, that's something that gets elided a lot in the reception of Darwin's work. And I do wonder how, I mean, you've written this, I haven't read them, I confess, but I've noticed and thought, wow, that's an amazing title. But I did read you one of the shorter papers on information, the Shannon Boltzmann Darwin, and in a sense, historically, that's a reversal, right? Because Darwin actually is an inspiration is an inspiration for Boltzmann, and Shannon, of course, is coming out of that, statistical mechanics way of thinking as well. And so, in some sense, Darwin and the way he treats variation is at the inception of the statistical turn in physics. But I mean, he's one of the main, statistics itself as a field is arising in that period as well, and that influences.
And it's not incidental that his father, the mathematician, was dealing with beginning of mathematical statistics. That was his field. And of course, Darwin being largely taught by his father, I think it's a part of that story as well. And so you're right that Shannon Boltzmann and Darwin actually reverses the historical tradition. But I think it's no surprise that Darwin's writing at the time when Maxwell is resolving these issues, when the idea of stochasticity is now finding its way into chemistry, into physics in a deep way. Darwin is right in his time in that respect.
100%, yeah. So he's a major, I guess, popularizer, at least, of that way of thinking, because his work obviously had such a big impact. But I feel that in a way, as we've discussed, that insight is sort of tamed in a way by its formalization by Boltzmann and others, right? So by the rendering of the fundamental state space, always predefined. And I feel like this is I know you've written a lot on the relationship between non-equilibrium thermodynamics and biology, but I think this is a fundamental limitation of those analogies, right? That those ways of thinking, and this would include, Prigogines, formalisms, and people like Hermann Haken and synergetics that come out of that whole lineage, which is kind of, I guess, still how a lot of complexity science wants to operate. Things like the free energy principle are still very much in that area. That there is this fundamental limit, like they can't really describe, it's kind of describing something that life does, but it's not going to be able to describe what life is, because life is generating novelty in the way that we are describing it, I think, but in a way that could never be, and I think not even in principle, right, reduced to that fundamentally predefined state space. It is not, that negative, purely negative conception where all the attractors in that state space already are there in the, fundamental postulate of the theory, essentially, the axioms. And then the system, like in Haken, order parameters can break down and it can move from one attractor to another, but there's no account, and it's not possible to give an account in those terms of the genesis of novel attractors, right? The genesis of novel non-equilibrium steady states. And that's why I think that analogy, it's an interesting analogy and it's a fecund analogy, but it will never be a homology, right?
Right. So, but this, of course, was what drove the writing for me of Incomplete Nature. First of all, incomplete is
Yeah.
what it's about. But the second thing is that I was reacting strongly to all of those people working in the area of synergetics or self-organization or far from equilibrium systems
Okay. Yeah.
who wanted to say that that's all we need.
Yeah.
That story is all we need to talk
Mhm.
about life.
Mhm.
So it's why in the middle of that book, I developed this idea that you need to have something like a near equilibrium thermodynamics process that I dubbed more generally homeodynamic processes.
Mhm.
Then you need to have the far from equilibrium systems are all processes that converge towards constrained dynamics and call that morphodynamics because it's producing form, regularity, reduction of constraint, excuse me, reduction of variety and increase in constraint.
Yeah. Yeah.
Then you need a third step,
Mhm.
and that's what I call teleodynamics. And arguing that the only way that that can happen is that if morphodynamic processes, self-organizing processes, each limit the destruction that they each will spontaneously derive towards. Self-organizing processes use up the gradient that drives them faster than they would otherwise. And so as a result, all self-organizing processes, all synergetic processes, according to
Yeah.
Haken, are in fact self-destructive.
Yeah. They leave no ancestors. So
That's right. So,
Yeah.
so that can't be the story for life. And yet living systems need to generate constraint
Yeah.
because they'll need the constraints to do work, but that work has to then regenerate those same
Yeah.
constraints.
Yeah. So yeah.
So what I realized is that the only way that can happen is by having complementary self-organizing processes, each create each other's boundary conditions. And what that means is that neither one of them can run to complete entropy. That is, they cannot use up you've got to have a system that blocks them from using this up. So I developed this process I call autogenesis to talk
Yeah.
about. That
I
is,
really enjoyed that paper. I misspoke, obviously, just a second ago. I meant leave no offspring, not they leave no ancestors.
yes, right, right.
Yeah,
No, yeah.
they're fundamentally dissipative, right? That's the point you're making, and that is the point of
That's right.
Prigogine.
But the key is that
Yeah.
those systems that do leave offspring have to use the dissipative logic, but they
Yeah.
have to use it against itself.
Yeah, no, I really love that insight, and obviously need to read Incomplete Nature. I've dipped a couple of times, but need to give it a proper
So
read.
I'll send you a short piece I wrote just
Okay,
about the thermodynamics,
that would
because
be great.
one of the
Yeah,
points I wanted to make is that we've got to stop talking about isolated thermodynamic systems.
yeah, yeah,
There are no isolated thermodynamic systems, number
yeah.
one. But in order to get it right, we need to have an even what I call the morphodynamics and
Mhm.
homeodynamics. We need to have a dynamical story that always works in open systems.
Yes.
You can actually look at an open system and look at a part of it and see that the part of it is actually eliminating constraints. Or you can look at a
Mhm.
part of it and see that it's generating constraints. So,
Mhm.
for example, the Bénard convection that produces, do you know what I mean by Bénard
Yeah,
convection? Yeah.
absolutely. Yeah.
If you look at just the dish of oil,
Mhm.
it's generating constraint, constantly generating constraint. But if you look at the oil in its environment giving off heat, it's of course
Mhm.
producing loss of constraint. It's
Mhm.
generating increasing entropy. So this
Yeah.
local, and this is a problem that comes with Prigogine is people talk about, is Prigogine talking about the reduction of entropy or the increase in entropy? Dissipative systems, they partition these two things.
Yeah, absolutely. Yeah. I mean, always the fixed boundary conditions, the broader system can be taken to be closed, right? In order for the local convection process, the Bénard cell, to export entropy to that larger system. And I mean, so it's still fully within the logic of closed systems, right? Which I guess is the point you're making. Yeah.
So,
No,
what I'm trying to do is to then change, to realize that the difference between what I call homeodynamic and morphodynamic systems is that to look at them and to analyze them, we artificially decide what are the boundaries that we'll look at. We bound it and we say within this region.
Yes. Yeah.
Within
So
this region. But the difference for what I call a teleodynamic system, like an organism, is that they actually create some of their own boundary conditions. They're not arbitrary boundary conditions, the result of an observer or an analyzer. They actually create their own boundary conditions, which is why they can reproduce themselves, why they can maintain themselves, why they can, in a sense, deal with degradation effects. And that's only because they produce their own boundary conditions. But the key is that self-organizing dynamics, because they generate constraint, are producing boundary conditions that something else can be bounded by. So it's only when you've got two systems that produce complementary boundary conditions and you find a way that they're coupled so that they each depend on the other to be generated. Now you can have a system that maintains its own boundary conditions. And so that's been my way of talking about what's simpler than life, but is that all life has to experience it.
Yeah, no, I think it's very well.
But I'll send you this short piece I wrote for a group called Thermodynamics 2.0.
Okay, yeah, I would love that. I'd love
it's
that.
a very short piece that just lays out the logic of how you would. I mean, I don't actually do it well, but I, in a sense, it's more like the formalism that preceded thermodynamics when we talk about increasing greater than or less than, not equations, but.
Yeah, I look forward to that. And I did read the autogenesis paper, and obviously we haven't really discussed that, but I enjoyed it very much. I think there would be a lot to go into in that discussion as well. But one of the things I really liked about it is the synergy.
Autogenesis paper?
The one you sent. So you emailed us one. It's from 2024, I think. What is it called again? An alternative path to molecular information.
That's right.
Yeah.
Talk about how RNA becomes information.
Yes. No, I appreciated that on a number of levels. I mean, I love I like the idea of the synergy first paradigm, which is really what you're talking about here, that there's a necessary complementarity of these processes. And so you could say in keeping with some of the discussion we've been having about biology, there's a necessary cooperation in a certain sense. It's a necessary ecology, right? And I think that helps us, which is again your point, to escape not just the non-equilibrium thermodynamics or the reductive utilization of that way of thinking, but the very related cybernetic way of thinking and the autopoietic way of thinking, like the kind of not sufficiently systematized or theorized notion of autonomy that exists in those lineages. And I sort of one of the things I like a lot about the work we've been discussing today is the notion of a sacrifice of autonomy. I mean, we didn't get into altruism and all of that much, but the way a sacrifice of autonomy leads to novel possibilities for evolution, but is a necessary feature of, I mean, I would say, and I think this is there in physics itself, in notions of frustration and that sort of thing, that you can't really have structure at all without relational co-stabilization. Right. So if there are things pushing against each other and holding each other in place, you would just have continuity, right? So there's a relationality to physicality itself, which I think is often overlooked in a lot of these discussions. Yeah, look, I loved a lot about the autogenesis paper. We probably don't have time or energy to discuss that now, but yeah, I really liked the analogy you have for horizontal gene transfer there. And so the utilization of, I guess it's like Bruce Deamer and colleagues. Is it Bruce Deamer? There's Damer and Deamer who've worked together, sometimes confusing with the names, but the wet, dry, hot, cold, or the other way around, cold, hot cycling, and the way that you've turned that into a theorization of cycling between closed and open states. And I think that, again, that's a beautiful way to kind of explode the ontologization of closure in all of this post-thermodynamic, but also cybernetic literature, showing like these two complementary operations. Yes, you need a degree of closure, but you also need openness for this kind of evolution to occur. And you can't dichotomize it in the way that Ashby does and then Varela. And, it's not just thermodynamic and operational. You can't dichotomize it that way. And I think there's still too much of that in the organizational literature even today, right?
And to some extent, the trouble with that literature is it's descriptive, but not very explanatory. It doesn't give you a process. It just says it has these qualities, these properties. And for someone who's, comes from biology, like you and I do,
Yeah.
it's just not satisfying.
Yeah, totally. So I reread last year for a cybernetics reading group the original Autopoiesis book, and I was so irritated by it. I mean, it's got a lot of stuff that I really want to agree with and a lot of beautiful stuff in it, but it's like exactly what you're saying. No explanation. The origin of autopoiesis is completely bracketed in that. It's like it's just autochthonous or whatever. It just
right.
I think one of the issues there is that they also wanted to their ambition was to replace evolutionary ways of thinking with an autopoietic, I don't know, postulate as a kind of the fundamental paradigm for biology. And I just, that kind of, I probably got my hackles raised from the very beginning. It's a bit of a manifesto, but yet almost no biological examples in the whole book.
That's right. Well, I think, and what's interesting is, of course, it was ultimately just a redo of Immanuel Kant.
Sure.
Yeah. So, you could read that whole thing in Kant's critique of teleological judgment. It was all there, this idea of mutually productive systems, and so on, using it to explain teleology. As a result, they just assumed it.
Yeah.
And of course, Kant recognized, what's interesting is that Kant recognized at the time that story couldn't explain it.
Yeah.
He ends the teleological judgment story by saying, there won't be a Newton of the blade of grass.
Yeah.
So I've always, similarly, I have a lot of conversations with these guys who don't see that difference, interestingly enough.
Yeah, I mean, there's almost a Fichtean sort of self-stipulation aspect, and it does trend very idealist, right? And I think this is also a problem. I feel like with a lot of the second order cybernetics literature, it's sometimes not clear. And again, I think this goes back to Ashby, that perturbations could be completely arbitrary in the sense that what defines the autonomy of the ultra-stable system, right, is that it will always respond to them according to its own internal logic, essentially, right? So they could be anything, but the organism will determine, in fact, what they are. So it makes it seem, it's the point you were making earlier, actually, that the stipulation of where the system boundaries are can be absolutely arbitrary. It's entirely on, in that. This is a problem with the free energy principle as well. Like, it's a great tool for systems identification. Maybe it's one of the best formal tools we have for systems identification, but it doesn't tell us how those boundaries come into existence themselves. We see them and we're like, well, empirically, it appears there's a boundary there. Okay, now I can draw a circle around it and I can talk about, I can talk quite rigorously, although again, with very few actual, well-worked biological examples, that's another issue, about what it would need to continue doing in order to maintain those boundaries. But the story about the genesis of the boundary itself ultimately becomes one, it's a story about a theorizer who draws circles around things.
That's right. That's right.
And I feel like that's there in a lot of the second order cybernetics literature, including even Bateson at times, right? So Bateson,
absolutely.
Yeah, he talks about maps of maps of maps. And okay, I get what he's saying. There's something interesting to be said there. But he says, he's talking to the Korzybski conference, and he says that nothing of the territory gets in. And I'm like, in that case, it's a pure idealism.
And the truth is that every map is a territory.
Indeed, exactly. Yeah, no, I love that. Beautifully put. Yeah.
Yeah. So the interesting thing is we have a lot of overlap. I like this a lot. And what's interesting about this is that it comes from biology.
Yes.
And for all the reasons that biology forces us out of simple straitjackets, there's always exceptions.
Yeah.
And yet there's also general rules but
Yeah.
they're not exceptionalist rules. They're in a sense tendencies.
Yeah. So it gets us out of that reductive way of thinking, right? Because again, yes, there are general principles, but they don't work in a reductive manner because you can't, there are always going to be exceptions. Not everything is explainable in light of that. Not everything is reducible to that. Yeah. No, I totally agree. I mean, I think my intuitions are totally formed by biology. I mean, including just spending a lot of my life walking around in nature and observing what organisms do. Like I always kind of wanted to be when I was a kid, a naturalist, not in the metaphysical sense necessarily, but in the kind of, Darwin, Wallace, like I was reading Malay
Oh yeah,
Archipelago.
yeah.
It's just like, just actually pay attention. And it's like organisms are doing all kinds of stuff, which again, if you don't ever use biological examples in your theorizing about biology, then you're going to miss all of that specificity. And it's going to give you the illusion that this formalism, this simple diagram, somehow everything can be reduced to it, which again, to your point, it doesn't mean that those diagrams aren't useful or even necessary at some level. It just means that they're not going to be explanatory of all specificity. And again, I think Peirce gets this. I love just what Peirce has to say about variation. There's a level of specificity which is not inexplicable. In fact, it's a precondition for the reality of explanation that
Yeah.
that you acknowledge a certain inexplicableness of a degree of spontaneity anyway. That's one of his intuitions and insights that I particularly value. Yeah. I mean, the other thing, and Parham and I talk about this a lot, is music for me. Like I'm a musician as well, like Parham. I'm not sure if you are, Terry, but that
Grew up in a musical family.
wonderful, yeah. So that engagement with music, both listening, but obviously playing and improvising, is also another, has shaped my intuitions a great deal. So yeah, I agree. We have tons of overlap. Obviously, you're like light years ahead of me in terms of the development of all of the theoretical framework. You've done
Well,
so.
I'm just older.
Well, I'm looking forward to reading a lot more of your work and hopefully having more of these conversations. And I would love to write something with you, just putting it out there if that's a possibility in the future as well.
Well, so let me just tell you what I'm currently working on.
Yep.
That is relevant, and there's multiple things, but there's one thing in particular that's relevant here, and that is what I call the transition from Fuka to Luca.
Okay. Yeah.
So Fuka is the first universal common ancestor.
Yeah.
Luca
Okay.
is the one that's got ribosomes.
The
And
last.
yeah, so
Yep.
the last universal common ancestor. But of course, Luca is incredibly complex. And one of the things that the paper you read is about what's the first step away from Fuka? Is how do you get to a system where now you can offload information onto the structure of something? And therefore, once it's onto a structure, you can copy it, you can use it again and again. That's really about, there's a piece I wrote before this a couple of years ago called How a Molecule Became a Sign.
Yep.
And it's basically the same claim. But my point is this, that argument ends, and you intimated at it when you were talking about the horizontal gene exchange,
Yep.
you might say on steroids in the case of this
Yep.
this case, in which you're exchanging all the possible combinable elements, the whole range. But basically, the Fuka to Luca story is, so how do you go from a system in which a molecule like RNA can both be linearized and use that linearized copyable sequence to create a three-dimensional structure that controls the catalytic activity between other molecules. So it's now information about a totally different molecular process. And that information is reproducible and translated into other contexts. But that's a very different information story than the genetic code information story.
Yeah. Yeah.
So this story requires that you start with something that's not like these weird molecules we call nucleic acids. Nucleic acids are just incredibly weird polymers. Why? Because they're made up of nucleotides, which are these, three totally different kinds of molecules stuck together, and then you make a polymer out of it. That's a totally weird system. So part of that story was to say, how do you get to something like that? But the next step is then how do you get to a system in which you've got a sequence that regulates catalysis? How do you get from that to one that produces the molecules, in this case peptides, that actually produces the peptides rather than depends upon them? So there's two parts to this story. One is, where do the first peptide-like molecules come from? I've spent a lot of time looking at hydrogen cyanide chemistry, because hydrogen cyanide, not on the Earth and not in water, but in other kind of solvents and other parts of the solar system, easily polymerizes. So hydrogen cyanide becomes a polymer in which the backbone is basically the same backbone that you see in a protein. HCCN, HCCN, CCN, CCN, CCN in this relationship. And I came to this by looking at the work of a couple of people that have followed the Miller-Urey experiment, and instead of looking at the water and what was dissolved in the water, looked at the sludge that was created, which turns out to be mostly hydrogen cyanide polymers. So if you first of all, hydrogen cyanide polymers are not easily miscible in water. They're not soluble, but slightly so. So what happens is they tend to break down. And when they break down, they break up into units that are basically like amino acids. So there's two sides to the story. Number one, I need to explain what precedes this autogenesis process, and how that comes about. The story I tell, that I have not really written up in any detail, is that hydrogen cyanide polymers are produced in huge quantities in the outer solar system, along with solid water, ice. But when dropped in on rocky, warmer planets where the water can melt and the hydrogen cyanide polymers can begin to break down, you get basically peptides of incredible variety. That's a starting point here. It's a variant of this same story, but at the level of chemistry. The second part of it is the Fukuta-Luka piece, which is, why is it that the whole system that's involved in Luka has so much redundancy? We think about the redundancy of ribosomal RNA. Ribosomal RNA and ribosomal proteins are produced in huge redundant numbers. The transfer RNA genes, there's 56 of them. You don't need that many. And the whole system is full of redundancy, genetic redundancy, incredible redundancy. Now, part of it has to do with the fact that you've got to produce maybe tens to hundreds of thousands of ribosomes in each cell and keep them going. So you got to have some redundancy, some, in a sense, dosage redundancy in order to keep up with that. But dosage redundancy is, of course, exactly the context in which this kind of inverse Darwinian process is likely to take place. In which there's error tolerance, where there's the possibility of driving this system. But if you have that kind of a system, and then you have this sort of hyper horizontal gene transfer effect, you now have the possibility of extreme sampling of these combinatorial relationships. You really have a great way to sample it in a non-lineage specific way. It's a population sampling, like the horizontal gene transfer trend has already suggested. People have done, I think, good simulations of this. But I think there's now a way to begin to use this logic to talk about how to go from FUCA, this very, very simple system in which you have an RNA molecule that can, in a sense, both be code and catalyst, to a system that has all these other complications. And what we've realized is that it's always by backing into it. The process is always the inverse of how we think about it in reverse engineering terms. So for example, how do you align catalysts so that they're in relationship to each other? If one catalyzes something that another catalyst needs as its raw material, how do you get them in order? Well, the answer is, if you've got an RNA molecule that produces a catalytic effect, you can offload that RNA molecule onto a larger linear molecule in which one follows another. The problem with linear RNA is it either folds up or breaks up. But if it can attach to a stiffer molecule, DNA, which instead of folding tends to produce these long strands of double strand that preserves the sequence and doesn't fold up. Now you've got a way to also line up and organize the catalytic structures, the RNA molecules. But it's backwards, it's thinking backwards from the way we normally think about it. So I basically, in that paper, I think you probably recognize this, by saying that in fact, the evolutionary sequence is inverse of the functional sequence. So it's not DNA first and proteins last. It's
Yeah.
proteins first.
Yeah,
Or
that's
peptides first.
yeah. No, I mean that is a basic insight that I fundamentally agree with, and it talked to my protein chemist colleagues are all believe that. Of course, it's just a bias of the field anyway. But
Yeah.
proteins are active. Yeah, look, there's so much there, Terrence. I mean, what some of the things that I'd love to read more of that detailed story when you've written it up and are willing to share it. But I'm thinking of some chemical ecology analogies, because in some sense this is kind of a chemical ecology type problem. And so, you've got discussion of how RNA scaffolding kind of works as an incipient canalizer of molecular interactions and that sort of stuff. And that's part of your story, right, about how information emerges as a kind of, it is replicable, but not merely a pattern that constrains other molecular interactions. And that, of course, is how, and this I'm sure, we didn't talk about biosemiotics today, and I'm not super familiar with that literature, but of course, that's in some sense my home base with all of these molecules that I used in a sign-like manner to constrain the states of, again, other systems endogenously, but also target systems in other organisms. And that even up to thinking of the DNA ribosome interaction in a chemical ecological term, the primary sequence is a constraint on the behavior of ribosomes that can make all kinds of things, but they are being constrained by another molecule to make a particular subset of the things that they can make. But one of the things that was coming to me when you were saying in this transition from Fuka to Luca and talking about all the redundancy is that I published a paper in 2016, which has a lot of other stuff in it, but it's called Rapid Radiations and the Race to Redundancy. And part of this was building on some earlier work that other colleagues of mine had done, but I was using it as a way of, I wouldn't say explaining so much as just talking about and conjecturing about evolutionary patterns that I was observing in the venom composition of Australian snakes. So I don't need to get into the details there, but there's this idea of a kind of two-speed evolutionary process where, and it had previously been observed in some invertebrate venom systems, and a lot of the invertebrate venom systems are far more ancient than the vertebrate ones. And what characterizes them often is that they seem to be very fixed. So they're at least anatomically, they're very fixed. The venom components can vary a lot, but these ancient lineages like spiders and scorpions and centipedes that have essentially had the same kind of venom delivery apparatus for 400 million years, as opposed to reptiles, which have evolved venom much more recently in the last 150 million years. And one of the really interesting things about reptile venom as a kind of model system for thinking about evolutionary problems is that the venom systems in reptiles, it's one particular lineage of lizards, which includes the snakes but is not limited to them, exist in all of these different anatomical arrangements such that you could kind of chart transitional forms even amongst the extant species. That's another discussion. But this notion of race to redundancy is the idea that, venom is a trait that trades on redundancy in multiple ways and kind of at multiple levels. One of the things we see a lot of, obviously, with venom, which is in that paper that you read, is that you see these gene families. Basically, one thing you can say is that a locus which has a propensity for duplication is seemingly more likely to be recruited, like its gene products from that locus are more likely to be recruited for a function in venom or perhaps in secretory systems in general. And that means you end up with this situation where you have redundancy at the level of the members of the gene families in a classic Ono style. And that's a great way of evading the possibility of resistance in target organisms as well, right? Like if a target organism only has a few copies of the target molecule, and you've got 60 in your toxin library there, like a drug library, a natural drug library, there's a good chance you're going to be able to stay ahead in that arms race, because you can overwhelm them with redundancy, you can overwhelm them with variation, because you've always got many, many different copies. And there what we see is that only 2 or 3 of those copies are probably highly expressed in the venom at any given time. Most of the copies are just sitting there, just constitutively expressed. So we infer they don't really have a functional role, except that they are grist for the mill of evolvability. But venom also has, venom as a secretion also has redundancy at a slightly higher level. Like say venoms are for predation or defense, or in parasitism, etc. Basically, feeding upon or deterring target animals. But say the whole function of a venom is to subdue a target organism, say for a predatory venom for a snake. But the venom can often do that in multiple different ways. So it'll have neurotoxins in there. It might have 2 or 3 different kinds of neurotoxins. So it's attacking nerve transmission at multiple levels. And then it might also have some hemotoxins and myotoxins and other things. And they often work on different timescales. Some snakes will kill target organisms, if they're rodents in particular, say, with hemotoxin very rapidly, but they also have incredibly potent neurotoxins, which take many hours to kill and probably are not part of the normal predatory dynamic. But they may have other functions like preventing the evolution of resistance as well. Anyway, the point is that there's all this redundancy in these systems. And so I conceptualized this race to redundancy, that in the lineages which had only recently developed a venom trait, that they were accumulating redundancy up to a certain level, and then subsequent to that, they would consolidate around a particular strategy. So redundancy kind of seems to accumulate early in the stages of evolution until there's a significantly broad repertoire, and specialization can often be secondary to that. I think it's very consistent with what you're saying, is that maybe in this transition from Fuka to Luca, what we're looking at is an accumulation of redundancy, which then enables a kind of launching pad in some sense. Like once you've got that much redundancy, you're able to do all of the unprecedentably vast variable number of things that living systems can do based on that, fundamental set of competencies.
Right. Yeah. Send me this paper.
Yeah, I will. Paper. Yeah, I'll send it to you with some caveats that I won't mention online, but
okay.
Yeah.
That's good.
Yeah.
That's good.
Great. It's been a really wonderful to chat.
All right. Yes, we should be in touch, and I'll share other stuff in your direction, and do it both ways.